Hello friends, we have started with discussion of plant hormone. We have already discussed about Auxin and Gibberellin Plant Hormone. You can refer my videos. The link is given in the description box. Today we are going to discuss about kinetin and cytokinin plant hormone. Here we will discuss about discovery of cytokinin, then its distribution and transport, then physiological effects of cytokinin, then biosynthesis of cytokinin, and at last we will discuss about cytokinin signaling pathway. So let us see them one by one. So first of all, discovery. Credit goes to Miller et al. In 1950. He was working on the growth of tobacco pith callus in culture. How it was discovered? So while working on tobacco pith callus, various growth substances, nutrients, vitamins, etc. were added into the culture medium, but these failed to cause any growth in the callus culture. Then they added content of old bottle of DNA present in the lab and observed growth of tobacco pith callus for longer periods. Later on, similar results were obtained with yeast extracts. But with fresh DNA, no positive results were seen. Therefore, they thought the active substance to be some degradation product of DNA. And they concluded that possibly it was purine compound. Later on, it was identified as furfuryl aminopurine. It was called kinetin because of specific effect on cytokinesis, that is cell division. But kinetin is still not isolated. Certain substances that show kinetin-like activity was isolated from a variety of higher plants collectively called cytokinin. These occur in plants, hence these occur naturally, and these regulate growth, hence called natural plant growth hormone. Some naturally occurring cytokinins are coconut milk factor, also known as liquid endosperm of coconut. These contain factors that show kinetin-like activity. Hence, these are used in plant tissue culture in vitro. Zeatin, chemical name 6,4-hydroxy-3-methyl-butrans-2-enyl-aminopurine. It was obtained as pure crystalline form from immature corn grains by Latham et al. Hence the name zeatin. It is highly reactive. Hydroxyl group is present in its side chain, hence it is more active. This is the structure of kinetin. This is the structure of zeatin. Distribution and transport. Cytokinin is found in transfer RNA preparations from a wide variety of organisms like brewer's yeast, bacteria, animals, and higher plants. So we can say that these are mostly found in all complex plants, bacteria, mosses, and fungi. Usually these are found in rapidly dividing tissues like root apices and growing shoot, etc. Therefore, present in highest concentration in these. These are synthesized in roots, especially during seedling stage, and translocated to shoots and leaves of the plant via xylem. Now, the physiological effects of cytokinin and kinetin. First is cell division. It induces cell division along with auxin. It is the most important physiological effect. Cell enlargement induces cell enlargement. Initiation of interfascicular cambium induces formation of interfascicular cambium. Morphogenesis has ability to cause morphogenetic changes in undifferentiated callus. That is when certain concentration of auxin and kinetin is added to the undifferentiated callus, it causes the differentiation or development of different plant parts. Example, high concentration of auxin and low concentration of carnitine results in development of roots. Low auxin concentration and high carnitine concentration results in the development of buds. Fifth is counteraction of apical dominance. We have already seen that auxin causes apical dominance and inhibit lateral bud. So in the culture, if auxin is added, then it results in apical dominance. That is, causes the growth of apical bud but inhibit lateral bud. And if the carnitine is added, then it counteracts the apical dominance. That is, it inhibits the growth of apical bud and allows the growth of lateral bud. But naturally, there is a balance between the growth of apical bud and lateral bud. And this is due to the balance in the concentration of endogenous cytokinin and indole acetic acid, which stimulate growth of lateral buds or control apical dominance. Now, the dormancy of the seeds. Dormancy of light-sensitive seeds can be broken by carnitine treatment in dark. So, light-sensitive seeds require light for breaking the dormancy or germination. But if carnitine is added, it overcomes the requirement of light and dormancy can be broken in the presence of dark. Similarly, carnitine treatment also overcome inhibitory effect of far red light. Far red light has inhibitory effect on seed germination, and if it is treated with carnitine, then it overcomes the inhibitory effect of far red light. Also, carnitine induces germination of seeds of parasitic plants in the absence of host. Parasitic plants require presence of host for the germination of seeds, but in the presence of carnitine, it overcomes the requirement of host for the germination. Seventh is delay senescence. Senescence, that is aging, can be delayed by carnitine treatment. It was showed by Richmond and Lang in 1957. Hence, it is known as Richmond-Lang effect. And how it is possible? So, carnitine treatment prevents degradative catabolic processes by repressing activity of few hydrolyzing enzymes like protease, RNase, DNase, etc. 8 is promotion of chloroplast development. When etiolated seedling is treated with cytokinin followed by illumination, that is exposed to light, then it promotes the conversion of etioplast to chloroplast. Chloroplasts then develop extensive grana and chlorophylls. 9th is anthocyanin synthesis. In many culture cells and tissues and in parts of intact plants, cytokinin treatment increases anthocyanin content because treatment of cytokinin promotes anthocyanin synthesis. And what are anthocyanins? These are flavonoid pigments which are responsible for red, pink, purple and blue colors in plants. 10th, other roles and effects. Some other roles and effects are differentiation of interfascicular cambium, lignification, accumulation of solutes, stimulation of several enzymes, especially photosynthetic ones. Now let us see biosynthesis of cytokinin. In the first pathway, we will discuss about how it is synthesized in plants. This is an important pathway and this is known as the IPMP dependent pathway. So let us see this pathway step by step. First of all, ATP or ADP combines with DMAPP, dimethyl allyl pyrophosphate, and gives isopentenyl ATP or isopentenyl ADP. This step is catalyzed by the enzyme isopentenyl transferase enzyme. Then this isopentenyl ATP or isopentenyl ADP is converted into isopentenyl AMP. It is then converted into isopentenyl adenosine by removal of phosphate group. Then isopentenyl adenosine is converted into isopentenyl adenine by hydrolytic removal of ribose group. Isopentenyl adenine is then oxidized to transzeatin by replacement of one hydrogen atom by an hydroxyl group in the methyl group of the isopentenyl side chain. This transzeatin is then isomerized into ciszeatin. This step is catalyzed by cis-trans isomerase. In this way, cytokinin is synthesized. Here, if we see the first step, ATP or ADP plus DMAPP is converted into isopentenyl ATP or isopentenyl ADP. This first step is the rate limiting step. We have already seen that this step is catalyzed by the enzyme isopentenyl transferase. Here, ATP, ADP or AMP is used as substrate and prenyl donor in this step is dimethyl allyl pyrophosphate DMAPP as discussed in the above pathway, or sometimes hydroxymethyl butynyl pyrophosphate, that is HMBPP. DMAPP and HMBPP that are used in the first step are produced by the methyl erythritol phosphate pathway, in short MEP pathway. So in addition to the above pathway, which is an important pathway for the biosynthesis of cytokinin in plants, cytokinin is also produced by recycled transfer RNA, which is seen in plants as well as in bacteria. It is not major pathway, it is indirect synthetic pathway. Let us see this pathway. Here DMAPP plus transfer RNA gives prenyl transfer RNA, and this step is catalyzed by the enzyme transfer RNA isopentyl transferase enzyme. Then cis-prenylation of prenyl transfer RNA takes place and it is converted into cis-prenyl transfer RNA. In the next step, transfer RNA degradation takes place and we get cis-zeatin riboside monophosphate. Cis-zeatin riboside monophosphate is then converted into cis-zeatin riboside, which is then converted into cis-zeatin. In this way, cytokinin is synthesized. Now let us discuss cytokinin signaling pathway. We already discussed various physiological effects of cytokinin. Example, it promotes cell division, cell enlargement, morphogenesis, delays senescence, chloroplast development, and many more. How cytokinin brings these effects? By the signaling pathway. Every signaling pathway has certain key components. Let us discuss them one by one. First is ligand, which is signaling molecule. Here it is cytokinin. Second is receptor, which binds with ligand. Example CRE1. In this video, we will discuss about cytokinin signaling in Arabidopsis plant. So here the receptor is AHKase. A stands for Arabidopsis, hybrid histidine protein kinase. Then third is transcription factor, AHP, histidine phototransfer protein. Fourth is inhibitor, type A, ARR, nuclear response regulators. Cytokinin signaling is two-component signaling system. It is of two types. One is very basic one, that is normal signaling system. It has two components, sensory histidine kinase and response regulator. Sensory histidine kinase is composed of input region and transmitter region, and response regulator is composed of receiver region and output region. Ligand comes and binds with the input region, which leads to autophosphorylation of transmitter region. This then phosphorylates receiver region, and output region of the response regulator leads to transcription of response gene. This type of the signaling system is seen in prokaryotes. Now, second type involves accessory protein in between. Like first type, it has hybrid histidine sensory kinase, which is composed of input region, histidine kinase region, and receiver domain, and response regulator, which consists of receiver region and output region. It also has an accessory protein. Here it is AHP. This AHP acts as a signaling shuttle between cytoplasm and nucleus in a cytokinin dependent manner. So first, ligand comes and binds with the input region, which leads to autophosphorylation of histidine kinase. Phosphate group is provided by ATP, which is then followed by phosphorylation of receiver domain, which then phosphorylates this AHP, which is followed by phosphorylation of receiver domain, then output region leads to transcription. In this way, signaling takes place. So here AHP mediate transfer of phosphoryl group from receiver domain of activated histidine kinase receptor to the receiver domain of response regulator, and this takes place in multi-step phosphorelay. That is, phosphate group is transferred in several steps in relay manner. So this second type of the signaling system is seen in higher plants. Let us discuss it in detail in Arabidopsis plant. So how cytokinin signaling takes place? As discussed earlier, it is phosphorelay signal transduction in response to cytokinin. Cytokinin, it is perceived or felt by cytokinin histidine kinase receptor. Then binding of cytokinin takes place to the cytokinin binding domain, which is known as the chase domain of the receptor. This binding activates dimerization of receptor, and it also leads to autophosphorylation on a histidine residue present in the transmitter domain, and this phosphate group is provided by ATP. So what happens? Ligand comes and binds with the receptor, which leads to autophosphorylation of histidine residue present in the transmitter domain. This is followed by phosphorylation of aspartate residue present in the receiver domain. Now, aspartate residue present in the receiver domain is phosphorylated. Subsequent transfer of phosphate to histidine residue on an AHP. Histidine phototransfer protein takes place, and this is followed by translocation of AHP to the nucleus. Then subsequently phosphate group is transferred to the AHP, and after the phosphorylation, AHP, which is present in the cytoplasm, is translocated to the nucleus. Then the phosphate group is transferred to the conserved aspartate residue present on the receiver domain of type B ARR. This leads to activation of type B ARR. Then phosphate group is transferred to the aspartate residue present in the receiver domain of the response regulator. Here it is type B ARR. This then activates type B ARR. This activated type B ARR then binds to elements within the promoter of type A RRs genes and other cytokinin inducible genes. This binding then leads to transcription of cytokinin primary response genes, including type A RR genes. This results in cytokinin response, which is seen as various physiological effects of cytokinin in plants. Type A ARR activate downstream proteins that are involved in cytokinin regulated processes. Also, it acts as feedback regulator and inhibit their own expression and cytokinin signaling. Now let us understand the cytokinin signaling with the help of this diagram. This is HK cytokinin receptor, which has extracytosylic region which is known as the chase domain, which binds with the cytokinin. Then transmembrane region is present and also cytosolic region is present, which consists of the kinase domain and the receiver domain. So first of all, cytokinin comes and binds with the chase domain, which activates the dimerization of the receptor and also leads to autophosphorylation of histidine residue present in the kinase domain. Then it is followed by the phosphorylation of aspartate residue present in the receiver domain. Subsequently, this phosphate group is transferred to the AHP, that is the histidine phototransfer protein, which acts as the signaling shuttle between the cytosol and the nucleus. This phosphorylated AHP is then translocated into nucleus, which then leads to the phosphorylation of the aspartate residue present in the receiver domain of type B ARR, that is the response regulator. This leads to the activation of the type B ARR. This activated type B ARR binds to the promoter region of the cytokinin response gene. This binding leads to the transcription of cytokinin response gene including type A ARR. This results in the cytokinin response, which is seen as the physiological effect of cytokinin in plants. Type A ARR activates the downstream proteins involved in cytokinin regulated processes. Also, this type ARR inhibits the translocation of phosphorylated AHP from cytosol to the nucleus, as a result of which type B ARR is not activated. Hence, the transcription of the cytokinin response genes does not take place, including the type A ARR gene. So, type A ARR regulates itself and its concentration. It is known as auto-regulation. So, this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.